Okay, good morning, everybody, and welcome. I'm Pat Suozzi. I'm going to turn my video on. I'm Pat Suozzi, President of the Federation of Vermont Lakes and Ponds, and I want to welcome you to another of our virtual events.
Pat, you must have hit the video twice because I can't
sorry.
see you now.
Here I am. You'd think we'd be perfect using Zoom by now, but you know, there's always glitches. So I just want to welcome you all to this event, Early Identification of Aquatic Invasive Species. I don't have to tell any of you how much of a threat AIS is to our lakes and ponds, and what a concern it is once AIS becomes established. The best method and the best defense against AIS, of course, is vigilance. And that's what this event is about, early identification and immediate action, the best ways to prevent established infestations. So I want to thank Jack, Widness, and the AIS, the FOVLAP AIS committee for organizing this, as it's a very important event and very important information. And welcome you all to this. So I'm going to turn this over to Jack, who's going to provide some details and introduce the rest of the program. So thank you, Jack.
Hi, glad to see so many familiar and so many new faces. As Pat said, I'm the chair of the FOVLAP Ad Hoc Aquatic Invasive Species and Water Quality Committee. For those of you who've never heard about us, we're just getting started this year. We're working today with the events committee to present what we have for you today. We're a new committee. We're always looking for input and new members. So if some of you are interested in doing the sorts of things that we're doing, which is working in a timely fashion with important things for lake associations, we really want to help you as best we can. One of the ways you can help us help you is to be by giving us feedback. And there's an opportunity for that at the very end of this morning or subsequently in terms of a short questionnaire that only take you a minute or two to fill out. But welcome. This has been a great group effort and collaborative effort. I want to acknowledge that this was the brainchild of Martha Winston, who's going to be moderating today's session. We think it's very timely for a presentation. In terms of a couple of housekeeping things, I would just ask you to please, while the speakers are talking, to mute your audio. If you have questions, please put them in the chat box. Beth Torpey, who's here today, will be monitoring the chat box and working with Martha to make sure that your questions get answered at the end of this. And as I mentioned, to the end of the day, we hope you'll fill out the questionnaire. So sit back, enjoy, think of the questions you have, and I'm going to pass this over to our MC and moderator, Martha. So Martha, take it away, please.
Okay, thank you. Good morning, everyone. We're going to get going right away. We are really thankful to have the person who knows the most about lake inspection programs here in Vermont, and that is Kim Jensen. She's with the Vermont DEC. She probably doesn't need much of an introduction. I think most people know who she is and have had trainings with her. She's going to talk today about the program, its history, what it does for the state of Vermont, how she hopes it can grow, and what kind of feedback they've had over the years to help them do early identification of AIS. So thank you, Kim. Kim's going to talk for about 15 or 20 minutes. Right after that, we'll have a Q&A. So like Jack said, as you listen, please type your questions in the chat box for Kim, and we will stop so she can answer your questions. Thank you. And Kim, it's all yours.
Kim, you're muted. Please unmute your.
I had such a great intro too. Thanks, Martha. It's good to see everybody this morning. Great to see some familiar faces as well. So I'm going to be talking for about 10 minutes, and then Carly, my AmeriCorps service member, is going to provide some great new news about Vipas. So I'm just going to share my screen, and hopefully I'll be able to show you everything I have to share with you about VIP. So Zoom is different for me. So let me just get myself organized here because I do Teams normally. There we go. All righty. So I think most of you that are here are probably familiar with the Vermont Invasive Patrollers program and been trained as a VIP, or as I like to say, a very important person. So I appreciate all that you've done, but I'm just going to go over, provide a brief overview of the program. And we have about 30 different VIP programs in Vermont. So this is a map of the Vermont Invasive Patrollers. The magnifying lens are those representative of those people. And the patrollers have mainly been for plant species, although that has not been limited to just plants. People find all sorts of aquatic invasive animals or just interesting things that they send me and share with me. So Vermont invasive patrollers are very important in Vermont because we have 800 lakes in Vermont, and I certainly can't get to all of them. This is a map of the infested areas in Vermont. The round red dot is Eurasian water milfoil. We only have one sighting of variable leaf water milfoil, and that was in Halls Lake, and I don't believe that is true anymore. I think that species died out, fortunately. We also have a lot of water chestnut in Vermont, spread throughout mainly the Champlain Basin, and then we have only a couple of instances of zebra mussels. But there's always new threats that are out there, and VIPs have been my eyes and Bob's eyes in the past for people just looking and making sure that they're keeping aware of any new infestations that are happening. And when they report things to us, we can quickly and hopefully get on any new introductions for a rapid response. Early detection is key to fighting any potential eradication for an invasive species in most situations. So this is a graph of the VIPs over the years. That has been going on for a long time, but we have records from 2007. And you can see in 2007, that's when Leslie Matthews was hired. She was hired to run the VIP program. And she actually would go out with people and do snorkeling with them. So I know that there's a bunch of folks that have gone out with Leslie and remember those good old times. We haven't been able to do that, unfortunately. And when the program started in 2007, we had over 80 participants. And as you can see, that population of trained citizens have decreased and increased, most likely due to staff, Vermont DEC staff, being able to train them. When I came on in 2017, I was hoping that would be my forte, and I could just take over that program and really reinitiate the program. But as many of you know, there was some challenges with staff leaving and so forth. So now it's down to one full-time person. Last year in 2020, the VIP program didn't happen due to COVID. We just didn't have the ability to train anyone or even to receive records. I think some folks did send in records, but I just was too overwhelmed with all the responsibilities of the program. But this past year, it was reinitiated with Julie Silverman, who came on. So she redeveloped some of the programs for us, and we were able to train. Now we have a great online presence, and we did have 60 VIPs submit their surveys this past year. So with our training, we have the VIP manual that's pictured here. And we also have a new key to common Vermont aquatic plant species. That's kind of the compendium to the manual. Before COVID, we actually did workshops, presentations in person, and we would also have some species that we would bring so folks could get used to keying out species and being familiar with the species and really seeing them up to close. And then we had a on-water portion where we're training people how to do a survey. And in most cases, we would start with doing a survey by everybody lining up, but then by the end, we are all kind of together in a squash of boats, looking at plants right next to each other. So that's always fun, having a flotilla of kayaks, trying to identify plants together in the field on the water. So due to COVID, we've tried to change that up. We now have online presentations. And last year we did 3 in-person sessions on the water. Most likely we'll follow that format again this year too. It makes it easier for a lot of people to get trained and then do in-person kayaks. So when you're preparing for a survey after all that training, we ask that you check out the lake scorecard. Most lakes have a lake scorecard. And if you go to the lake scorecard, you can see down here, there's a little pie chart. And if it's red, that means that you have an invasive species in your water body. And at the lake scorecard, there's also this view plant list. And you can click on that. And if your lake has been visited by a botanist from the state, we have a plant list already available for you, so you know which species should be in your lake. So you print out that lake list, and then you can also print out a picture of your lake water body too, and prepare yourself for going into the field by familiarizing yourself with what plants should be there. Once you're gathering all that data, you also want to take a look at our plant survey data sheet. And so there are particular things that we hope to gather, such as where you surveyed, what's the latitude or longitude of the sites, when did you go out, how many people went out with you, how many hours, and then of course what the conditions were, and then what you may have found. You could also put in any native species. I know a lot of people who've been doing this for a long time do like to list native species for me. And then you have a list of different equipment and supplies that you should go and bring with you. So once you're out in the field, if you're doing it with others, it is helpful for you to kind of line up and either do a parallel survey, and that's what this is showing. You can see the tracks in the duckweed, where every line was covered. And we also have aqua viewers that folks can use. That's what these are. These are big culvert piping, and it has plexiglass at the end so you can see down into the water. So we have some of these available for folks. And then once you find something, you can pick it up and try to identify it.
Thank you for the seminar.
And then once you have completed your survey,
We
then
need people to mute their speakers if they're not talking directly. So please mute your speakers.
so once you get back on shore, you can kind of do a map of the lake and what you covered, and then just a quick list of all the species you may have found in there. And you can also map kind of the big communities like water lily or lodia or Eurasian water milfoil. So those big assemblages of plants, you can map that out. This is Eden's map. So they also provide me a map of which section, which VIP had surveyed. And then they have a concurrent data sheet that matches up with those areas. And that is sent in to me. And then also, if anyone has any aquatic specimens, submissions that to send in to me, or now if they could take a photograph of it and send it to me, that's even better because I'm no longer in the office. That's another thing that's available for folks. So I have the pleasure of finding and identifying a lot of species for people. A couple of VIPs have had some quick saves throughout the years. Some have found Eurasian water milfoil. A VIP up in Magog found starry stonewort for us. And we were able to get on those infestations pretty quickly. The starry stonewort, it's unfortunate that there's not a control program for starry stonewort, but at least now we know that it's up there due to a VIP, Valerie Dillon. So that is the program in a nutshell. I'm going to share my screen, or actually allow Carly Alpert, who's the Eco-AmeriCorps person, to share her screen.
Kim, we have one question in the chat for you.
Sure.
The question is, with almost 800 lakes and about 200 with Eurasian milfoil, what is the rate of transmission of Eurasian milfoil to uninfested Vermont lakes?
It's a really good question. We have 100 water bodies with Eurasian water milfoil. The transmission is pretty low, actually, which is amazing to me. I think a lot of the Eurasian water milfoil was introduced in the early 90s, and then it kind of probably was a wave. I actually should do a graph of that. That would be really interesting. But recently, there hasn't been any new introductions except for Salem, unfortunately. But I think because of the Greeter program and VIPs, we really have not had new introductions of Eurasian water milfoil in my time, at least, or at least the past 5 years. We have had some new introductions. We had 11 new rapid response events last year. So someone found something new and we quickly went and did some surveying, and we only found 3 new species in new water bodies, and 2 of those were water chestnut and 1 was brittle naiad, but not a Eurasian water milfoil. Good question. So I'd like to introduce Carly Alpert, who is my Eco-AmeriCorps person, and she's been working on our new program, a new community science program called the Vermont Invasive Patrollers for Animals program. So Carly, hopefully you'll be able to share your screen.
Yes. Can everyone see my screen?
Looks good.
Perfect. Okay. Hi, everyone. Like Kim said, my name's Carly. I'm the Eco-AmeriCorps member working at the Aquatic Invasive Species team at the DEC. And I'm really excited to share with you our new community science project that we've been working on. It is called Vermont Invasive Patrollers for Animals, or VIPA for short. It stems from the VIP program, but like the name says, rather than focusing on aquatic invasive plants, it focuses on aquatic invasive animals. And Kim and I felt that this program was necessary because Vermont doesn't have an official list of aquatic invasive animals. So this distribution data that our volunteers would be gathering for us would be really important and helpful in monitoring and preventing new infestations. I will give you the roll call of the animals that we're including in the program. We have the Asian clam, quagga mussel, and zebra mussel for our bivalves, the big water crayfish, rusty crayfish, spiny and fishhook water fleas are our crustaceans, banded and Chinese mystery snails, and faucet snails are gastropods. For fish, we've decided to include the alewife, European rudd, northern snakehead, and the round goby. The round goby is really interesting and important because it's been getting a lot of attention lately. It's been discovered in the Champlain canals, so it's going to be especially important that volunteers are trained in identifying this fish as an introduction in Lake Champlain is very possible. Finally, we have one turtle as part of the program, and this is the red-eared slider. So if you decide to become a Vipa, then you will have access to our training manual. You'll be given a Vipa kit, and you will be required or asked to attend 2 workshops. One will be online, and it's kind of like the background information, survey techniques, identification skills, and like why the program matters. And then the second training is going to be in the field. And this is where you learn actually how specifically to survey for these animals, how to use all the equipment in your Vipa kit, that sort of thing. But everything that you would need to be a Vipa will be provided for you in your kit. There is an additional option if you would like to be a zebra mussel monitor or a crayfish monitor. You would be required to hang a sampler on your dock or have waterfront property where you could put a trap. But again, this program is really meant to be super accessible for all Vermonters, and we encourage everyone to participate. You don't need to have a dock. You don't need waterfront property. You just need this kit, which would be provided for you, and the willingness to walk along your local water body and survey for these animals. So now I'm going to show you some of these resources that I've been talking about. This is our brand new Vermont Invasive Patrollers for Animals website. And here you can find all the information that I've kind of been talking about. You can access this website from the DEC website. It's also linked on the Vermont Invasive Patrollers website, which maybe you are more familiar with and know how to get to. I'm also happy to drop the link in the chat, so that might be the easiest. This outlines the responsibilities of a Vipa, what we would go over in the workshops. There's also the link to the Vipa Manual, which I talked about briefly, and this really is going to be your go-to guide for being a Vipa. It includes survey pages. Well, I'll just show briefly. It has identification pages for each of these animals that I was talking about. This is meant to be just quickly help you identify the animal in the field. It also includes survey directions, and these are all materials that would be provided for you in your Vipa kit. And it also includes field sheets, because some people might be more comfortable with a paper field sheet, so that if you don't feel comfortable using your smartphone or bringing your computer out in the field to keep track of your data. In addition, I'm just going to quickly show how we would like for people to be submitting their data. We're going to be using iNaturalist as well as a web form, which will be published in June. So iNaturalist is a really cool application. If you are not familiar with it, or if you don't have an account, that's totally okay. It's really easy to sign up. You just enter your email, username, and password, and then you can create an account. I already have an account, so I'll just show you quickly what it looks like. Oh. to make an observation. You would just go, to add observation right here. And then you would choose a file. Hopefully you're able to upload a photograph to iNaturalist. And then it would ask you for a couple more questions about like your location and the date that you had this observation. And then it actually will compile your observation into the Vermont Invasive Patroller for Animals Vipa project, which I'll show you really quickly. Vermont Invasive Patrollers for Animals project. And this will keep track of all the observations of these animals across the state. So you'll be able to see what other Vipas are observing. And it just kind of serves as a network for volunteers. But I'm not gonna spend too much more time talking about iNaturalist because I saw that Julia will be talking later more about this application, which will be super cool. Yeah, and that's all I have to say about the program right now. I think Kim and I now will be happy to answer any of your questions about either program.
Thanks, Carly.
Thank you.
Okay, we have some, oh, are you looking at the chat, Kim?
Yes.
Okay, good.
So there's no way, we also have a VIP webpage. Hopefully you're all familiar with that. I can share the VIP page with you. Okay, here we go. So we have the VIP page, and on the bottom is all the different resources, the data sheets, the training manual, the survey data field sheet. So we have an online data sheet now as well. So you could submit your surveys online. And in the question, there was, is there a way to upload your photos? And unfortunately, we cannot upload any documents to the ANR webpage. It's just not able to be done because of security purposes. So you could send me your photos or your data sheets in an email. It's fine to do so. So I think the other question was, Beth, if you wouldn't mind helping with me, there's another question there.
Okay, just a second. It is a question about the risk of wake boats in with AIS because of the ballast, since the ballast can't be emptied completely or inspected.
Yeah, so the water, normally folks should be taking their ballast water out from the lake that they're coming from, because they're not going to be driving with thousands of water in their boat and trailering that. The amount of water that is left in a ballast tank is small, and if it was traveling over land for a few hours, let's say, let's hope even overnight, then it's highly likely within 24 hours or within 6 hours actually that any kind of larvae villagers would be destroyed. So there's a great paper that was done that I can share with the group that was done that shows that villagers do not and cannot sustain within ballast water for a long period of time because it gets too hot and they basically die. It's not saying that all of them don't die within a few hours, but it's highly unlikely that ballast water would be a case of a new introduction.
Okay. We also have, what are some examples of problems or the negative effects of specific invasive animals? And that could be a really long answer.
Yeah, so basically exotic invasive animals cause great economic and environmental harm. They can wipe out an entire zooplankton food web. such as the zebra mussels, that's what they do. Quagga mussels do the same. So there's not food for the native mussels. They can also severely impact the native population by taking over their habitat. Same is true with zebra mussels and quagga mussels. And it can also clog intake pipes. So if you have a water source coming from a lake that's infested with zebra mussels or quagga mussels, it would clog those intake pipes. So there's a lot of great information that will be in the Vipa manual, so you can look up any resources and references in that manual for more information.
Okay, thank you, Kim. There's one more, and that is, what do you do about the invasive animals systematically once you find them?
Some things there's not much we can do, such as zebra mussels. We are fortunate that we have calcium low waters in Vermont. So there's only 2 established populations of zebra mussels in Vermont, in Lake Bomazene and Lake Champlain. They definitely do have an impact on the native mussels, but we haven't seen them making an impact on the economic infrastructure. However, other species that may be able to get introduced into Vermont water, such as round goby, which is in the Lake Champlain Basin area in New York, in the Hudson River, that could have some serious impacts on our fisheries and our tourism monies that come in, because it may impact the bass populations and other fishery sports, tourism dollars. So there's a lot of impacts. Each specific animal has its own particular impact. And again, I would ask you to check out Carly's new Vipa manual to learn more about those animals.
That manual looks great. Great job. There's a couple more questions. When and where can we find out about the training dates for both these programs, the VIP and the Vipa?
I was hoping that I would have those today, but I unfortunately had so much to do. So we hope within the next couple of weeks, we'll have those posted on the webpage.
Okay, and one more from Marco. He says, Kim, I was not clear, or him or her, what AIS were you talking about in terms of life in a ballast, and what study did you reference?
So the ballast water is can contain zebra mussel veligers or quagga mussel veligers or any other kind of microscopic or macroscopic invasive species, such as water fleas too. So those types of organisms can be sucked up into the water, or even in live wells or other water compartments. And so those are the types of invasive animals we're talking about. And I have a study, I don't remember the title, but I can put it in the chat box as others are speaking today.
Thanks, Kim. I actually had a question. I remember there was a study a while back saying that the veligers of the zebra mussel could survive up to 30 days. So I assume that the study that you're referencing is a newer study?
There's many studies, some studies look at favorable conditions. These were looking at realistic conditions. So they were actually testing ballast water. So yeah.
Great, thank you.
Mm-hmm.
And that's it for the chat.
Great. Well, we still have 1 minute left for Kim or for Carly. So if anybody has something, type quickly. I see one more coming in, I think, and then we're going to move on to our guest from New Hampshire. Do you see one more, Beth?
Yeah, in order of significance, once you find EWM, what have you found the most efficient means of control? So if we find Eurasian water milfoil immediately, the best thing is to dash it or diver operate suction harvest it out And if that doesn't work, then bottom barriers are the next line of defense.
And Amy also has a comment that the indications are that water temperatures over 140 degrees Fahrenheit are needed to kill invasive aquatic animals.
And that's generally what decon stations are. But if you have a heated decon station, they heat up to 140 degrees. That's not true for ballast water. It may not heat up to that temperature, but the fact that there's not going to be oxygen in the water, or if it's in there for a long period of time, then most likely the organisms will die. So two different types of heating mechanisms and environmental conditions that we're discussing here.
Okay. Thank you. Kim, thank you very much.
You're
I
welcome.
know I'm hoping most of these people here will see you on a training this spring, and we also hope to get back to paddling. I know that. And thanks to Carly for starting the new program. I hope you get some recruits today.
You too. Thank you so much.
Yeah.
Yeah,
All right.
Thank you.
We are going to move on because we have a guest from New Hampshire. She does kind of what Kim does here in Vermont. She works with the exotic species, and she's the coordinator of that program. Her name is Amy Smegula. And thank you, Amy, for being with us. We will probably learn some new things that we can do in Vermont as maybe they do them in a different way, or even a better way in New Hampshire. We'll find out.
All right, thank you for that introduction. And hello, everyone, our neighbors to the west. As I was introduced, my name is Amy Smagula, and I've been with the Department of Environmental Services in New Hampshire for 24 years and counting at this point, I believe. And yeah, I've been coordinating the invasive species program for that entire time, as well as doing water quality monitoring and other lake-centric activities. So I'm going to talk a little bit about New Hampshire's program today and draw some parallels to what Kim and Carly were talking about. Sort of behind the scenes, all of us, Kim and I and our counterparts throughout the Northeast, are all active in the Northeast Aquatic Nuisance Species Panel, excuse me, among other groups. So we have the opportunity to share and learn from each other and mooch from each other. So you might find that our programs and our content are very similar. But I will add some insights as to how I work with volunteers and recruit volunteers in New Hampshire as well. All right, just trying to advance slides. There we go. All right, so New Hampshire's approach with aquatic invasive species, I'll briefly go over that, and then I'll talk about the specifics of our Weed Watcher program that we have in New Hampshire. So the elements of our program are, we try to be as comprehensive as possible. We do a lot of prevention activities, signage, laws, rules. We have a program also for lake boat launch stewards that we call the Lake Host program that does a lot of our prevention activities. And then our monitoring for early detection is through our network of volunteers, but also our biologists that go out and do surveys. We control infestations. We do research for new control techniques or into the biology and ecology of species. And then we do a lot of that regional collaboration and also national collaboration so that we are competitive in terms of, you know, keeping up with the current technologies and methodologies with other states. So you have an idea of what's going on next door to you. This is the state of New Hampshire. We unfortunately have a little bit more diversity in invasive species over here. We have a lot of different species that we're tracking. Our problem is not as much Eurasian water milfoil like it is for you, but we have more variable milfoil here than we have Eurasian. So all those red dots are all variable milfoil across New Hampshire. The green dots and some of the blue dots are Eurasian water milfoil. We also have fanwort in southern New Hampshire. We have a couple water chestnut populations over on the Connecticut River and down in the Nashua River. And then a smattering of other species across the state, Brazilian elodea, a few spiny naiads or European naiad. We've got curly leaf pondweed in a few water bodies. We do have Asian clam that we're tracking. This is actually up to 6 water bodies now. This map is slightly out of date. And then we have a whole lot of Chinese mystery snail across the state that isn't represented by dots on the map because we don't have a good handle of where they are exactly. Oddly enough, our infestations are mostly in the southern half of our state. We have not yet documented anything in water bodies in the northern half of New Hampshire. And I know you guys have populations of infestations in the northern part of your state. So I'm just not sure why we don't have any yet. I'm not complaining, but it's weird. If you are interested in learning about any water bodies in New Hampshire, we have a DES Lake Mapper app. You can just go into Google and type in NHDES Lake Mapper as shown here, or when you get the slideshow, you can just click on the URL here. But it brings up a map of the state and you can zoom in on a water body. Here I zoomed in on our border water, the Connecticut River, and you can find reports, management plans. You can see the list of species that we've documented in a water body that are invasive, and a whole bunch of other information. So this is our online app for water bodies in New Hampshire. And I stuck this slide in here when somebody asked the question about the rate of spread of species. So this is data from our New Hampshire Lake Coast program, so our boat steward program. And I'll just explain it quickly. Over on the vertical axis over here, this is the number of inspections that are done each year. Over on the right, this is representing either the number of saves or the number of boat ramps staffed. So in New Hampshire, we built up to staff about 100 of our high use boat launches a year. And those are done on the weekends or sometimes during the week. So this is about 100 boat launches. This is about 100,000 or more since about 2015, 100,000 or more actual inspections done in the state of boats that are coming in and out of lakes at boat launches. And then the orange bars here are the number of saves. So a save is when you are actually intercepting something that's on a boat or trailer that's launching or coming out of a lake. So you can see we did have, as we increased our program number of inspections, we did have a really big rise in the number of the intercepted species or the saves of invasive species on boats and trailers over the course of several years. And then as we continue to increase our contact with boaters and educate boaters, we did start to see a steady decline in the number of species that were moving around the state. So of all these different inspections, I probably looked at about 300 plants over the last few years, so 300 each year. And of those, only a small percentage were invasive plants or animals that were moving from boat launch to boat launch. So that just gives you an idea of, as we stepped up our education and outreach activities, we actually did see a drop in the rate of spread, which is encouraging. We're really glad to see that. All right, so some things about our Weed Watcher program. One of the things that we understand well and that we've learned over the years is that flexibility is key. The state of New Hampshire provides a framework for a Weed Watcher program, and we want to make sure that it fits all groups. Some lake associations are much more active and engaged than others. Some lake associations have 2 or 3 people, some have 50 people. So the range among lake associations is quite varied. So that framework is applied to each lake association or volunteer group, and they make it their own based on how much they can do. So they tweak it as appropriate. And then we build in the flexibility based on how we train, how they do surveys. So that could be by boats. Kim mentioned snorkeling. We do that here too. Paddle boards, paddle boats, canoes, kayaks, whatever gets people out on the water. And then how people report varies as well. So we try to keep it loose, but also provide a framework so that people have an idea of how to go about doing this. We've actually had volunteers across a range of groups. Most are lake residents, and a lot of those folks have been, a lot of you are going to know this, they've been doing this for many years. A lot of them are retired, and they have a lot of varying interests and activities that they do, and they really hope that new people coming to the lake volunteer to help, but it always seems to be the same couple of people that are stuck doing everything. They organize the meetings, they do the weed watching, they do the water quality sampling, and they're looking for more people. So we reached out to a bunch of groups and tried to build interest in local lakes among people that might not live on lakes or ponds. So we reached out to local high schools and colleges and advertised the program and requested volunteers. And students always need volunteer hours. So that's a good way to bring people in and educate them. We've also had a lot of interest from Boy Scout and Girl Scout groups that learn these plants and animals, and they can earn badges for projects. And the interesting thing is that a lot of invasive species are found by kids. A lot of zebra mussel infestations and other things across the country have been found by kids. They're very visual, and they see these things and they'll say, "What's this?" So they're good groups to target. We've also had a lot of interest from local garden clubs who actually do a lot of work on land, but want to take their volunteer time into the water. So they've been a source of good volunteers. And then a lot of fishermen's groups have had interest in keeping an eye out for invasives on their favorite water body. And they often reach out for information. And then teachers who have summers off like to have different activities to do either on their own or bringing their kids out. So they are also good sources of volunteers too. So if you have a lake group that's struggling with volunteers, these are some options for you. A similar map to what Kim showed, and actually full disclosure, I mooched this from Kim, the idea for this type of map. So this map is showing the distribution of the volunteer weed watcher program folks that we have across New Hampshire. We have a big gap in this part of the state that I need to work on, but we do have a fair number of volunteers across the state. So we really haven't had a hard time getting programs going. So a lot of active folks out there. The program's been in place for several decades now, and it does still continue to grow. And as Kim said, there's a lot more to look for than just plants. We may need to rename our program beyond Weed Watcher these days because people are looking for invasive plants and animals, and they're also looking for cyanobacteria blooms or blue-green algae blooms these days. One of the things that I always have to remind volunteers is not to get discouraged if they're not finding anything. That's really what we're hoping for. But the whole purpose to look is just to make sure you don't have anything. Or if you do, then report it. Another thing too is that lakes that do already have an invasive species in it get discouraged as well. But I'm seeing more and more water bodies in New Hampshire where we have more than one invasive. We have some water bodies that recently just got 2 or 3 new invasives in them, or some that historically have had as many as 6. So it's always good to keep looking even though you already have something. Our trainings are done in various ways. So like I said, we try to meet the needs of these groups. This is a much younger version of me back in the day when I was doing a pontoon boat training on Crystal Lake in Eaton, kind of close by you guys. So we go out and train groups. Either I'll go out or I'll have my interns go out and do trainings. They can be on the water. They could be virtual, especially nowadays, or we can do in-person trainings. There we go. So we do try to adapt to the needs of the group at hand. The trainings are conducted every time a new group forms, and then every couple of years, if a refresher is needed, we'll go out and do a refresher, but we don't have the capacity to train every year for the same groups. And lately, like you guys are doing right now, we also record the trainings and have them available on YouTube so that people can go in and either rewatch it or share it with their new volunteers so that they can become trained easily and effectively in the comfort of their own home. And let's see. So local program structure, like I said before, we try to be very flexible. We do tend to have a lead contact on everybody's water body. So that could be a head weed watcher, if you will. But there are some groups that it's everyone for themselves. They don't have a formal structure, and we don't try to force that. Reporting, I know you guys, like Kim said, have a more formal reporting process online. In New Hampshire, it's a little looser than that. I usually tell folks, let me know if you found something and you think it's bad, but some groups report every time they go out and they survey. So I'm fine with whatever is being reported as long as folks are out looking. And our recommended frequency for surveys is once a month from May through September as a minimum guideline, just because invasives can grow so quickly, but more frequently is welcome. Some folks weed watch every time they go out on the water, which is great. I know I do. It's sort of an occupational hazard, but less frequently is not desirable, but we'll take it. So if somebody only looks, we have some folks that come to New Hampshire for a week, a summer, and that's it. And they'll look when they're there. So at least it's eyes on the water. So I wouldn't say don't start a program if you don't think you can meet a rigorous schedule or requirement. Flexibility is really important. Our equipment needs that we typically recommend, any type of vessel is useful, but you don't need a vessel. You can wade, you could snorkel, scuba dive, whatever works for you. We do recommend that if you have a motorboat, that you have a driver and one or more observers. We do have that Lake Mapper app. People can get the lake outline map and depth maps, which are useful, and historic plant maps on there, and that they can take those out on the water body. We don't ask everyone to survey for all plants and all invasive plants or animals. We just say, you know, become familiar with what should be there, and then look for anything new or different. And then I'll talk about that more in a minute. We provide plant and animal identification keys, which I'll show you in a minute. We recommend that folks have long handled rakes so they can reach down and grab something off the bottom, or a throw rake, which is a double-headed rake on a rope. The Siver screen is new for the last few years. We actually do have an invasive animal monitoring program specifically. This is a high-end stainless steel sieve that we use, but honestly, you can just use like the kids' sand toy sieves, which are the plastic ones that are super easy to find in the local stores this time of year. But for the animals, we recommend wading out to about knee deep. These are my interns. And then pushing the sieve down into the sediment by about 6 inches in the lake bottom, and then bringing it up to see if you have any Asian clam, like this guy right here, or Chinese mystery snails, or any smaller animals like that. So that's quick and easy to do, and we have methods for that. And then we have our volunteers out topside doing surveys for plants and other things. We always recommend having some type of Ziploc bag with them. We actually created bags in New Hampshire specifically for the Weed Watcher program. I'll send a picture of that so that you guys can have access to it. But it's printed on one side with collection methodology, and on the back side, it has a form for you to fill out with water body name, contact information, and location of where you found what you found. So it's a specific bag for that. And then we recommend polarized glasses and view scopes like Kim talked about. The polarized glasses are really useful for cutting the glare on the surface and seeing in much better. And then on the water, we recommend breaking the lake up into different zones so that not one person feels responsible for the entire water body. We know that that can be pretty off-putting when you feel like you have to survey the whole lake and be responsible for the whole lake. So we find this zoned approach works better, remembering to include any islands that are on your water body. And then also, when you're surveying, we recommend covering as much area from shore out to where you can't see bottom anymore, doing some type of parallel pattern or zigzag pattern offshore, and then also doing the same around the islands, just so that you can see as much area as possible. And then while you're out there, scan the surface, scan the water column, scan the bottom, and see if there are any invasive plants or animals. And then I always tell volunteers, you're looking for anything that's new or different or out of place that wasn't there last month, last year. You guys are the experts on your lake. You've had history there, you notice those changes. Whereas Kim and I or Carly, when we go out to these lakes, maybe once a year or once every few years, don't have that nuanced knowledge of the lake. So you guys will definitely notice changes first. I've had volunteers find single stems of new plant growth in their lake that's been invasive at the single stem level, which is pretty remarkable. Anything that appears to be growing quickly or taking over, anything bright green for plants. A lot of invasive plants tend to be a much brighter green color. Animals or things that look like they're covering the bottom in high number are all good things to look for. Anything new, different, or out of place. And then some tips for going out. Try to go out on calm days or low boat days when the water is smooth, so not too many waves or ripples. And also trying to go out when the sun is high, so the water column is lit. And then all areas of the lake where sunlight reaches the bottom, so from shore out to where you can't see bottom anymore. And that's where your scuba divers and your snorkelers will come in handy. You might want to put them out in the deeper zones and have people in boats in the near shore zone where they can see better. And then more frequent surveys may be needed at the outlets at the windward side of the lake. So if you're doing a training, just ask who gets everything when the wind blows. That would be that shoreline. Any boat launches and areas of historic dredging. Dredging is a disturbance, and invasive species really like disturbed areas. And then in New Hampshire, we recommend that if you find something, mark it so that we can go back and find it again, so that if you have to call Kim, she knows where to go. So a buoy, a GPS waypoint, triangulating where that spot is, flagging the shoreline, and then any other notes you can make on that site. And then in New Hampshire, we do like voucher specimens, just like in Vermont. So we ask that volunteers collect a voucher specimen. If it's a plant, representative material of that plant. If it's an animal, if you could just collect one, that's good, because sometimes, especially with things like Asian clam, there are endangered fingernail clams that look a lot like them. So we want to make sure that we don't wipe out a population by collecting too many, just for ID purposes. And then we've also found, and this is a pandemic age thing, that instead of having samples dropped off at our labs, we do prefer that digital photos come in. And we always recommend placing that specimen on a white background and arranging it out so it can be seen clearly, and then putting some type of scaled item next to it so we can see how big it is, and then taking a digital picture and emailing that. I know Kim and I both check email regularly, so our turnaround time is pretty quick with identifications in our respective states. Like I said, online trainings are really useful because they can be recorded and saved and revisited. Another thing that I do in New Hampshire is I or my interns will take a lake that we're training and we'll create a personalized slideshow for them. So we will go to our plant list and any other notes that we have on the water body, and we'll create a slideshow with photographs of everything that we know that should be there. And then we'll include pictures of all the things that you don't want, all the invasive plants and animals, and then save that. And with the goal of hoping that the volunteers will review that, become familiar with what should be there, and then sort of set a filter as they're out on the water body and ignore the stuff that should be there, and then hone in more on the things that are new or different or that aren't in their slideshow, and then have those things checked. So it's just another reference or resource.
Yeah. Amy, we
Yeah.
have a lot of things in the chat for you.
Okay.
So can we, are you okay with moving on to that?
Yep, I have two more
Oh,
slides. I just want to
okay.
show you. Yep.
Thank you.
So I have laminated cards. I know Vermont also does. They're good resources. And then you guys can take a look at the publications that will be listed when you get the slideshow. So go for it.
Okay, we do have a bunch in the chat. The first thing's a comment, not a question. Chrissy Cano wanted to shout out to Shadow Lake for early detection and then eradication of milfoil. Thank DEC and Kim. Next question is, when you find a new invasive species in a water body, can you give examples of where this information has been effective in either eradicating or better managing the new invasive?
Yeah, absolutely. So having a regular weed watcher program out there, or even just lake residents looking in their lake regularly, helps tremendously, because in the past, when people weren't looking, we were finding things on the multi-acre scale. At that point, it's really hard to manage. So like I said, having volunteers out looking regularly, we can find things in the single stem or small patch area. Then all we have to do is send divers in to hand remove it, and it could potentially be gone. So just like your variable milfoil infestation in Halls Lake in Vermont, that was found early, it was dived on, and Kim thinks it's been eradicated or it's waned. So having that really helps us get ahead of an infestation, and we can manage it much better. And eradication is actually a word that might be in play at that point.
And Kim says that Shadow Lake is a great example of that. Mark Malassa wants to know about underwater cameras, and David Wieselmann responded that GoPros work great, and we'll be seeing some of that in the Seymour Lake presentation.
Yeah, we're starting to have a lot more volunteers using underwater cameras and sending us stills from those videos for identification. So that's really useful. It lets you cover bigger areas that you might not be able to see easily from topside. So yeah, we are encouraging camera use, either stills or video.
Chrissy also wanted to point out that Leslie Matthews indicated that algae can grow up to 30 feet deep. And then David Johnson wanted to point out the significant differences between the New Hampshire and Vermont programs. How many staff are part of the New Hampshire program?
Yep. So to the Christine's point about Leslie's observations, we've seen variable milfoil growing down 30 feet deep in Squam Lake, which is exceptionally clear and colorless. So they can grow deep. So having those, and you can't see it from the top. So having your scuba divers or your snorkelers that can go down a little bit deeper and then, come back up and, report is helpful. The differences between Vermont and New Hampshire, as you saw from Kim's talk and my talk, are not that different. I am the only person in the invasive species program regularly. I have half a biologist who helps out from time to time, and then I have seasonal interns. So it's very similar to Vermont, and we try to maximize our capacity by bringing volunteers in to be the eyes out there. It's very helpful to all of us.
Great. One more question. Jack Widness wants to know if you use DNA analysis of AIS to confirm your findings, especially with so few saves in New Hampshire in recent years.
Yes, yeah, we actually do use DNA quite a bit. A lot of the milfoil species are really hard to differentiate between. And what we're seeing now a lot is that some of the Elodias, or the water weeds, are very similar looking and hard to decipher. And with hydrilla, a looming threat, we're really trying to make sure that we're positive on plant ID. So we've been doing a lot more DNA in recent years. And we've been doing a lot of eDNA too.
I was actually going to ask about hydrilla. Do you have any detected in the Connecticut?
No, we have not found it yet between Vermont and New Hampshire. The closest place to us is southern Massachusetts down by Agawam, and we don't have any in New Hampshire. The closest spot to us is Pickerel Pond in Limerick, Maine, which is western Maine, about 8 miles from the New Hampshire border, but we believe that that's been eradicated.
Oh, great. One more question. We're right on the edge here. Do you have any experience with Crocella core in milfoil control?
Yes, I do. I have widespread experience. We started experimenting with Crocella core in 2016 in a small limited trial. And then when it was labeled for use, we started using it in 2018. And that was just a few lakes. In the last 3 years since it was licensed, we have moved up to having used it in 50 lakes across New Hampshire. And I've found that it's really effective on variable milfoil and Eurasian water milfoil, and it's a reduced risk herbicide. So I've found that we don't have as many non-target impacts as other products we've used, and it's gone from the water column in 24 to 48 hours, which is much better than any product we've ever used before.
Great, we're over right now, but Chrissy has a question about hybrid milfoil. The species detected are hybrids of the aggressive variable leaf milfoil, the Eurasian milfoil and variable leaf.
Yep, good question. So Eurasian and variable haven't been shown to hybridize yet. A variable milfoil and Eurasian water milfoil hybridize with other native milfoil species. And we have not found any hybrids yet in New Hampshire. Kim can speak to what's been found in Vermont.
Great, thanks.
Okay, Amy, thank you. That was so fascinating, and your slides are going to be reviewed a lot of times here in Vermont, I think. So we can, take some of the things that work for you and hopefully use them here. What a great resource you are. Thank you so much for being here today. And we are going to move right along. We have one more speaker, and then we're going to be taking a coffee break, and then we'll have a little roundtable discussion after that. So our third speaker today is Julia Pupko. She is a community science outreach person with Eco AmeriCorps, and she is an expert on using the iNaturalist app for identifying aquatic plants and other plants, and also Seek, which is another app. So she'll be giving some training to us a little bit. And if you have iNaturalist on your phone, you might want to, train along with her. Okay, thank you. Julie, are you ready?
Yep.
Okay.
All right. Hello, everyone. I'm happy to be here with all of you today. Carly actually gave a really great introduction to the content that I'm going to be talking about today. So without further ado, let's talk about iNaturalist. So to give a little more detail as to what iNaturalist is and what it does, it is an entirely free platform that connects people with science and allows users to provide crowdsourced identifications. iNaturalist provides a really great platform for naturalists of all skill levels and interest to come together and learn while also contributing to research. And one of the benefits about iNaturalist is there is a browser version, which I will talk about first. There is an app that you can download on your phone. They have both an iPhone version and an Android or other smartphone version. One thing to note is that those two apps, the one for Apple iPhones and then the one for other smartphones, are slightly different in how they function. And that can be a little bit confusing, but there are a lot of great resources out there, hopefully myself included, to help you kind of navigate that. And finally, there is this app called Seek, which is a little bit separate from iNaturalist, but you can connect it to your iNaturalist account. And I will get into that more in a couple minutes. So one of the best parts of iNaturalist is it allows for communication and collaboration between researchers, community naturalists, and people across everywhere, and enables for data collection on a really large scale. Because it is a free app, tapping into community volunteers or being a community volunteer yourself and using iNaturalist allows you to very easily increase community engagement with research in a really fun and educational way, raises awareness in a very low cost manner, and increases the chances of finding target species, whether those are invasives or native species that are not doing so hot. Additionally, there have been some studies that have shown that community science contributions, this was for a specific lady beetle study, but the community science contributions were correct over 81% of the time. So using the verification or identification component of iNaturalist can really allow you to get some pretty high quality data for whatever that project may be. So moving right along, the basic unit of iNaturalist is this thing called an observation, which you can see right here. Carly very nicely showed how you get to the point where you are uploading these observations. And basically each observation should be of one organism at one given time. So say you are observing an aquatic plant that is perennial, and you take a picture of it this year, and then next year you go back and you know it's the same plant, and you take a picture of it again. You would not want to add those new photos from the second year to your original observation because it is at a different time. You would instead want to create a new observation. This allows you to kind of track individuals over time as well, which is pretty neat. So I am going to jump to the website really quick and show you a couple of the different features here. Everyone can still see my screen, right? Okay, great. So as discussed with uploading, one of the benefits with the browser version is that you can upload multiple observations at once. So I'll back up and talk through what I just did. You can either click add observations here or this green upload in the upper right-hand corner. Choose your files, and you can actually batch upload a bunch of images at once. And how you do that is you single click on the first observation you want to upload, hold shift down on your keyboard, scroll down to the last one you want to upload, single click on it and click open. You now have a bunch of photos. Each photo comes up on its own. I call these plates. But as you can see here, I have different plates that represent images of the same organism. And I want to group these together because I don't want to artificially inflate the population of this one organism by uploading multiple observations of it. So how you combine, you single click on the first, hold shift down on your keyboard, single click on the last image that you want to combine, and click combine up here. Another thing you can do, so you see this guy here is the same as this guy down here. You can just click on the corner and drag. And now you are able to combine the rest of these that are all nicely next to each other and get your observations all together. I'm just super quickly going to say here, so iNaturalist has artificial intelligence. You can see just by single clicking on the species name, this is indeed a Hyperascus lady beetle. Apologies for all the beetle examples here. I'll talk about aquatic plants in a little bit. So you can either just click on one of the iNaturalist suggestions if that feels correct, or if you know what this species is, you can type in the name. Oops, if I can spell, T-A-V. There we go. And then you want to make sure that you have an accurate date. It's okay if you're not entirely sure. You're like, ooh, I know this was in April, but I'm not sure what day. You're still going to have to enter an exact day and then just make a note down here. And you also need an accurate location. So you can either type in latitude and longitude, search for a location, or zoom in on the map and drop a pin somewhere. All right, so after you've sorted all of these, you just simply click submit. I've already added these to iNaturalist, so I'm not going to do that. But a couple other features I wanted to talk about super quickly. These are easiest to find when you are on the browser version versus the app. but they can be seen in both. You have an Explore tab here, which allows you to search for species within a specific location, view them as a grid, a list, or on a map. And you can add additional filters to better curate your search. Filters button here, more filters option here. You type in everything you want. So say you want aquatic plants, you would type in, or like say milfoil, you want milfoil, you type that in up here within a certain region, add a couple more filters and click update search. Another thing to note is that there are taxon pages. So this is going to take me to an observation, but if I click at the name at the top, you get a bunch of information about this species, and you can also do the same search for larger taxonomic groups. Here you can get a map, which you can further filter using filter by place up here. You get about information that links to Wikipedia, taxonomy, status. It'll also tell you if something is introduced down here, and similar species, which is really helpful for identification because you can see what other species have been misidentified as the species of interest. The final thing I'm going to talk about right now is you do have the Identify tab, where you can go and help other users identify different species. Same filter situation as the Explore page. And then there's some really nice ways where you can click through this, but that gets a little bit outside of the context of this video, but I'm happy to answer questions on that. So popping back to my presentation here. All right, let me get this back in full screen. There we go. Great. So super briefly on the mobile app, I'm just going to talk about how you would upload an observation here. As I said, it has very similar features to the browser version, not quite as many, but pretty comparable. And it's not quite as easy to navigate through as the browser version. So even if you only have a smartphone, I'd recommend just typing iNaturalist into your internet if you want to explore the full situation of iNaturalist. So to upload an observation on the mobile app, after you download it, you click Observe here. You can either add a photo from your camera roll, or directly take a picture. One thing, I recommend taking photos of the organism outside of iNaturalist first, and then adding photos from your camera roll, because if you just add it directly from the app, you can only take one photo at a time. That's less of an issue for plants because they don't go anywhere, but when you're trying to maybe document an invasive beetle or something, aquatic beetle, they move, and it can be tricky if you can only take one photo at a time to actually capture a good photo. Then from here, you have the same, after uploading your photos, you have the same information here where you get some AI information about what you see, and then enter in all the rest of your information. Another thing to note is that you can change the settings on the app so it does not upload automatically to iNaturalist, and instead will wait until you are at a place where you want to upload it. So if you're in the field and don't have service, you don't have to slow down iNaturalist by automatically uploading those photos, which is pretty nice. So another option that we have is Seek. This is a really great way to get kids into identifying things because it's sort of one of those point and identify apps where you can point your camera at something. It'll give you suggestions as to what the plant or other organism is. And then you can capture a photo and connect Seek to your iNaturalist account. So you can upload photos directly from Seek to iNaturalist. Seek also has these really fun challenges. So it'll be like, how many tree species or how many invasive aquatic plant species can you capture and identify in a month. So again, this is a really fun way to get kids or families into using iNaturalist and identifying things. So trying to be mindful of time here, just going to move through a couple more things. Another really fun aspect of iNaturalist is the ability to create projects. So what you can do is on the iNaturalist top banner, you can see on this left picture, there's this community tab, and under the community dropdown, there is an option called projects. So you would go there and set up a project. I'm just going to talk about collection projects right now. Basically what those do is, remember those filters I talked about on the Explore page? When you set up a collection project, you select a series of filters, and every time the homepage of your project is loaded, iNaturalist runs a search, grabs all of the observations that fit those requirements and pull them into the project. So this is a really great way. You can see here, there's this Maidstone Lake project set up by Chris Von Alt. Any observation that is uploaded within the boundary of Maidstone Lake will be pulled into this collection project. So it makes it a really easy way to view all of the, in this case, aquatic plant observations within that boundary. So really quickly, just going to talk a little bit about how this connects to research. Once observations on iNaturalist have been verified, which means that they have become research grade. In other words, other users have come in and either confirmed the original identification of the species or have been like, hey, this isn't a milfoil plant, this is instead whatever. From there, once there's 2/3 of community agreement, it'll become research grade and it gets pulled into these larger databases such as GBIF and the Vermont Atlas of Life Data Explorer, which is something through the Vermont Center of Eco Studies where I'm serving currently. And from here, there's all these great ways where you can view and map your data that allows you to really easily download data from a bunch of different sources. So both from things like iNaturalist, where it's crowdsourced and has been verified, and also from any research or organizations that have done a Atlas project or something like that, and then have digitized their findings. All of that information you can get for a certain location during a certain time frame and any other filters that you may want. So this is a really awesome way for both researchers and community naturalists to have access to a bunch of open access data, view it on maps, and download it as necessary for further analysis for whatever projects you may be contributing to or running. And then finally, I'm going to talk a little bit about observation qualities. Observation quality considerations. So if you're not taking photos that can be identified by other users, you won't be able to get your observations to research grade, which it still can be a very valuable observation, but it isn't going to be as valuable to research if that is what your goal is. So you want to make sure that you're photographing multiple parts of the organism, getting both close-up shots and far-away shots, especially for plants, have an accurate time, date, and location for when that observation was taken. And when we're talking about plants specifically, you want to be thinking about, first of all, where is the plant growing? If you can get a shot including the plant in its habitat. So if it's an aquatic, is it growing out of the water? Is it kind of on the shore where there was water in the spring, but as the summer progresses and things dry out, it's now, you know, kind of like a terrestrial plant. Is it fully submerged in the water? So get a shot of that. And then you want to think about the leaf architecture. Does it have simple leaves or compound leaves? There we go. What do the leaf margins look like? Is it sinuate, where it's kind of like this flowy, ripply situation? Dentate, where it kind of looks like a bunch of little teeth. Sorry, these photos are much blurrier on the full screen than they were on the tiny photos. Or is the leaf margin entirely smooth? And then another thing you want to photograph if possible is the leaf venation. Is it really parallel? So all of the veins in the leaf you can see right here are parallel to each other. Pinnate, which means that the main veins here kind of look like the pinna on a feather. Or is it kind of a fan-shaped leaf with all of the veins stemming out from one main point? And that concludes my presentation. There are some links which I can pop in the chat, but you will also be receiving this presentation in an email. So you can follow these links and feel free to reach out to me with any further questions or help setting up your iNaturalist project or getting started in iNaturalist. Thank you.
That was great. Thank you, Julia. We don't really have questions, but some comments about using iNaturalist to set up a project and then using Seek for the local homeschool group. That sounds like a lot of fun. And I think that's it. Everyone says thank you and great job.
Awesome. How much time is there left in my section? Because I'd be happy if folks want to talk a little bit more about projects. Like I can pull up the Maidstone Lake project and kind of talk about how you set that up and things like that.
Okay. Yeah, Julia, we were going to break at 10:35, which you've got about 6 minutes if you would like to. I think the projects are probably where a lot of lake associations would like to go if they have people technologically able to. So, okay.
Sure. Yeah.
Thanks.
And just a clarification while you're bringing that up, for those of you interested in today's slides, they will be available shortly on a link on the FOVLAP site. So don't expect an email, but go to the FOVLAP website and you'll be able to get the presentations perhaps in a day or two.
Great. Thank you for that clarification. Okay. So here is our project page. Again, you go community, drop down to projects. And to start a collection project or any other project, you simply click start a project here. So one thing to note is that you have a couple different options here. Again, collection projects are kind of the easiest to manage because iNaturalist just filters observations using, again, similar filters to the Explore page. You see you have your taxa, your places, users, and a bunch of other information here. You do also have the option to set up an umbrella project. So when you would use this is when you have, say, projects for, I don't know, 4 different lakes within a watershed, and you have a different collection project for each lake, and you want to see how all of those projects are doing at once, you can put together an umbrella project, which would be like a home landing page with links to each of those projects. And then you can see the statistics for each of those projects kind of compared to each other. So that can be fun if you want to set up like a BioBlitz or something and sort of have a fun competition between different lakes or watersheds or whatever the case may be. You also do have the option to set up a traditional project. I'm not going to get into much detail on that other than it is much more difficult to manage because people have to do a secondary step of uploading their observations to that project. It doesn't get automatically pulled in, but you can make those traditional projects a little bit more niche. It has like more filtering capacity than your collection project does. Another thing I wanted to note is when you are setting up a collection project and want to do it for a specific place, in Vermont, counties and towns can all automatically added as places. In other states, the smallest level that iNaturalist automatically has is the county level. And there are also some natural areas that have either been added by other iNaturalist users or have been automatically added when iNaturalist was built. That said, there are a lot of lakes that don't have a boundary in iNaturalist already. So what you would have to do at that point before you could type in your place name here would be going to more and places. And then you would scroll down to add a new place. And what you would have to do is upload a KML file of the boundary of whatever lake it is. Places do slow down iNaturalist a bit. So make sure that you're keeping the size of the place the smallest that you possibly can. So don't make a place that's like every single lake across the United States. Keep it to like a lake or maybe 2 lakes. And then the other thing is the searching mechanism here for places isn't the greatest, but you really want to make sure that you are searching places first to make sure that somebody else hasn't already created that place before you go to make your project. And now I will just quickly pull up the Maidstone Lake project. You can see here there is a place added first to get Maidstone Lake into iNaturalist, get that nice lake boundary into iNaturalist, and then was used to create the project. And you can see here we have this beautiful project home landing page. You get a nice overview of the current statistics, so the number of observations that have been uploaded, number of species found, number of identifiers, and number of observers. And if you scroll down a little bit further, you get recent observations, another breakdown of the statistics, and you get information on the project requirements here, another breakdown of stats, which you can click this to see different breakdowns of the organisms, and this will also take you to the explore page so you can filter things a little better. And I see we're running up on time, so I'll just be quick here. You have your boundary with the map and points here. You can click on each of these to see what the observations are, and then click to navigate over to it. Then the final thing is you have a journal option. So what you can do is after people join your project up here, I'm already a member, so you don't see the join option up there. But when you create a journal post, it will get sent out to the people who have joined your project as long as they select that they want to receive notifications. So that can be a fun outreach tool right in iNaturalist.
That's awesome. Thank you so much, Julia. And one of the questions was about showing Maidstone, which you did. And the other question is if you're willing to help individual lake associations get going for an iNaturalist project on their lake, if you can help remotely or if you need to be there in person.
Yep, so I'm happy to do that. That can be done remotely. It's honestly much easier to do that remotely than in person because we can be on Zoom and I can just share my screen and talk you through the process as we're doing it. And my position at VCE is primarily supposed to help community naturalists use iNaturalist and other crowdsourced platforms. So definitely here, I'll pop my email in the chat right now. Definitely
Perfect.
feel free to reach me out or reach out to me with any questions, and the resources that will be uploaded with this presentation will be very helpful in terms of getting you started.
That's great, thank you. And then another question, what about where there is no cell signal? That's where Seymour Lake, there's very little cell signal there. If we're out on the lake and we want to take a picture of an underwater plant, what do you do? I think that's what you meant, David.
So that is totally okay. If you don't have like if your phone location isn't working at all because of data, just kind of like note relatively where you are. You can adjust the size when you're uploading those observations later to be the total potential area where you viewed that organism. So that's totally okay. Also with using the mobile app, again, you can take photos in the app or put observations into the app without cell service and upload them when you get back to a place where you have reception or internet. Just make sure if you're using the mobile app that you go into the settings and make sure that you turn off the automatic upload to iNaturalist in the mobile app. Otherwise, it will try to upload those as you finish the observations, and it just like freaks out if you don't have internet service. So just make sure you turn that off. It's right in settings.
That is good to know, thank you. And that is it for questions.
Right. Julia, thank you. We might have to do a whole seminar just on this. It's fascinating. And like I said, I know a lot of people are going to be wanting to add this to their toolbox for their aquatic invasive species and their lake. We're going to go ahead and break. We have a coffee break planned, and we're going to shorten up just a little bit because I don't want to get up too far off schedule. I want to be able to finish up by noon. So if you will plan to be back to start the roundtable at 10:50, that gives us all about a 12-minute break or so. And I hope you're interested in the next section. It's going to be 4 speakers telling about their lake and what they do on their lake with their VIP programs and their experience and history. And hopefully you'll learn something if you're starting a program, or learn how to improve your program if you already have one running. Okay, thank you to our morning speakers. If they're leaving, I'm not sure. I'm assuming they probably are. Kim, Carly, Amy, and Julia, you guys were really wonderful. You brought us so much great information. Thank you so much. We'll see you in about 12 minutes. Okay, all right. This is Martha Winston again, and I'd like to welcome you back. This portion of our presentation is going to be a roundtable discussion. We have 4 people representing 4 different lakes or ponds. They're each going to take about 10 minutes to tell us about their program. And then at the end of the 40 minutes, we will have an open Q&A for all those people who shared. So if you, again, go to your chat box, if you have questions, you can put the person's name that's sharing so we know who to ask, or if it's just a question in general. All right, and we're starting off with David Wieselmann, and he is at Seymour Lake. David, are you ready?
We're starting out with me.
I'm sorry, Beth. Beth is at Seymour Lake too.
To David. My name is Beth Torpey, and I'm the program manager for the Aquatic Invasive Species Prevention Program at Seymour Lake, which includes the patroller option. And Seymour Lake is big. It's one of the largest lakes fully within the borders of Vermont. The original native name was Namagonic. That means salmon trout spearing place. I thought that was pretty cool. And now, the first AIS grant was written in 1999 by me and the president of the Seymour Lake Association at the time. And we included provisions for the lake bottom monitoring, or the patrolling. Andrew Emery was the first patroller, and he's in the picture here with his kids. He may have been the first VIBP patroller in the state. We originally just had one person. He was also the greeter at the time. But then when Janet Selby took over, she developed the current framework, which David is going to get into. And David Wieselmann was hired in 2017 to coordinate the patrollers and to develop the lake bottom map. And now David's on.
Hello, everyone. Great meeting today. I'm so excited to use the apps that Julius discussed earlier. That's going to be a game changer. We do have a humongous lake. It's about 11.7 to 9 miles around. So we have a lot of volunteers. We have 12 to 13 volunteers spread out around the lake. And each one of those volunteers do have family members that help them. So a lot of those do have 2 and 3 sets of eyes on these areas. We're really lucky because all of our VIPs have grown up on the lake, multi-generational, and really are passionate about keeping our lake invasive free. We do a couple different patrols. Well, first off, when we do first hire or bring on a VIP, they're trained, which of course there's multiple training sessions throughout the summer. We were kind of put in the back seat with COVID, and we did some virtual training, and I also provided different videos for people for milfoil and zebra mussels and different things that we're dealing with in our area, not in our lake, fortunately. So we do a patrol. Basically, we try to get people to go out in the beginning of June, and then preferably around the end of August, even early September if possible. Last year, our lake was pretty cold up until mid-June. So we noticed that there was a lot, our densities were a lot smaller than previous summers, which is great. So that's beneficial. We also all have some of the cleanest water in Vermont, but we do have a huge job in front of us every year. We have a massive lake. It's up to 160 feet deep, which 65 feet deep, we don't have to worry about that, but we have huge sandbars, and we have rocky areas. We have different types of areas that we have to patrol. Of course, since last year, we have the online submitting, which is great. And we do take note of how many hours people patrol every summer. And also what I've built is kind of an interactive map that shows where our patrollers live, what their areas are, and then there's a report of what they found. As someone discussed earlier, we're basically looking for zebra mussels, which is north of us, and also, of course, milfoil. We have different techniques for patrolling, and I love this about our lake. Everyone goes at it in different ways. I kayak. We have people that canoe, jet ski. Kenny Janess, she's amazing. She'll snorkel for miles. And this is great because she goes way beyond her patrol area, and her and I keep in contact multiple times throughout the summer. She's amazing. So she kayaks and snorkels. And if we have any questionable areas, which is like our boat launch areas, we really can get somebody underwater to take a look at those really challenging areas. I personally, because I was in a mountain biking accident 10 years ago, I use a underwater GoPro camera, and 2 of them. And so what I'm able to do is I'm visually looking, of course, but if I have any questionable areas, I can put my GoPro camera underneath it, under the water. And we can take a look at it. I can freeze frame it when I get home on my laptop. And I can also, this gives me an exact geolocation of this areas, which is so necessary for such a massive lake. So we are able to find, if we have any questionable areas. I do bring a rake. I do take samples with a bag and a rake, but if it's a little deeper than I'm able to go, I can get Kenny to go take a sample, or we can take a look at it on my laptop. I know we don't have much time here, but this is kind of just showing where people live, what their patrol areas are. You can see this is a massive lake, and we have an inlet and outlet that we also have to patrol. So it's just short of 12 miles, which I also, just because I have a lot of time on my hands, I'm just kidding. I patrol the entire lake as well into like literally September, October until I leave. So we're very fortunate to have a great team. I'm looking for more people, more the merrier. If anyone has any questions about the GoPro camera after this, please email me, reach out to me. We were so privileged to have some time with Ann Bove a few years ago. She's been at this patrolling for 20 plus years, I think, maybe more. So knowledgeable. Beth Torpey, myself, and Ann went out, I think August or late July, and did a patrol. We showed her what we're doing, and we actually, well, she found a new species on Seymour. It's called quillwort. It's non-invasive, but it is new to our lake. And so we're able to, with the camera, pinpoint it. And real quickly, since we found it, it has grown considerably on our lake. Real quickly here, our wish list for this summer is more VIPs. We do have a huge Eastern coast that we need a few more VIPs, and the more the merrier. My goal is to have a GoPro camera connected to an iPad so I'm able to look directly under my boat. And as well as that, I've reached out to a couple different companies about underwater mapping software with the drone. However, Julie has kind of opened my eyes on different options, excuse me, on what we can do. So I'm really excited about using that. If anyone wants and has any questions, please reach out to Beth or myself.
And I wanted to say the secret sauce of our program is connection and community. We have generations of stewards, as David saw before, and there's a lot of people that are really connected to this place.
And I hope I didn't take too much time. I know we're in a rush here.
No, you guys are within your time. Thank you.
Okay,
And Beth and David have been kind enough to share their emails. So I know you guys would take email questions at home, right? At any time?
Absolutely.
Okay.
Definitely.
Okay. And this will be in the information that FOVLAP will have with all the presenters' information. So, yep, we hoping to build a network of experts, right? Citizen scientists.
Yeah.
So thank you, David. Thanks, Beth. The next person who's going to be at the roundtable is Jeri Bergdahl. And Jeri is on South Pond. And Jeri, do you feel ready to go? Don't forget to unmute yourself.
Hello, everyone. This is the view of South Pond in Eden, where I've patrolled since 2007. And I think Kim said that was the beginning of the VIP program. And I remember that as the beginning. This is a July view. Then this is a fall view of South Pond. And why I wanted to show this slide was because you can see that the shoreline rises up quite quickly into the trees, so that the lake becomes deep fairly soon after the shoreline. So it is, but it's a beautiful, beautiful spot, an oligotrophic lake. South Pond is about 1 mile long and approximately 120 acres, but you can see there's a lot of shoreline here for only one person to survey. And luckily this year, this past couple of years, I've had a couple of other people join me. There's not that many camps on the lake, and many people are, gone just there for the weekend or whatever. So there's not many people that have been willing to put time at the lake into surveying. So what I say is take time to get to know your lake. It's really important. I heard that a couple of times from Amy and from Kim. Know what's in your lake and the immediate surroundings. And the only way to do that is to keep looking. And I attend training sessions whenever possible and look online or look at reference sources as often as I can. And another point that was made was find the list of plants already identified on your lake and then try to find them. I was very excited when I found the last one just a couple of years ago. There was just one plant I've ever found of that particular plant that was identified earlier. And then ask DEC, Department of Environmental Conservation personnel to confirm your findings. And I was out with Leslie Matthews, and it was a joy to be with her to confirm some of the things that I had found on our lake. But I check every single time I'm on the water for what is on the lake. A useful tool for viewing the lake is the viewing tube. And you've seen people using this tube today. It's about 3 feet long and about 10 inches in diameter. And the one end is covered with the plexiglass and sealed. And I find that I can store this in the hull of my kayak, which makes it available in the kayak. It seems like it might be too big, but I can get it in the hull in my kayak. This is another view through the plexiglass to the open end. And this was taken just recently as I wanted for this slideshow. So you can see our lake is still frozen. But something you might view is this freshwater sponge. And I heard, I think it was Amy say something about bright green things. When I saw this for the first time, this bright green thing, I was just flabbergasted. I thought, oh no, what do we have? And it turns out to be a freshwater sponge, which is indeed an indication of a clean freshwater lake. While surveying your lake, I like to carry a covered jar. I've heard other people say plastic bags, but to conserve the integrity of the plant, I use a jar. That's to keep the plant really hydrated. And a kayak paddle actually is pretty useful for dislodging plants, but it kind of moves you around a little bit when you use it, but it can be used. Rake is better. Silt covers live plant materials in certain parts of the lake. So be sure that you sort of swish that silt off, because you'll be surprised what you find. I was really surprised. Aquatic moss oftentimes gets covered with silt. And of course, GPS is best, but I'm reluctant to take my phone out too often when I'm in the kayak, because I always think, well, it's going to go overboard. So I look at the shore, try to find a place, something that identifies a spot, looking in the water for a rock or sticks or something, and then use that as best I can. One important thing is to greet fellow lake owners when you're out there. Tell them what you're doing and what you found. It builds community. It's really important to do that. The next time I'm around and I'm poking around their shoreline, they wonder what I'm doing. They oftentimes will say, "Oh, hi, Jeri, what are you finding?" And that's great for community. Back at camp with the plant samples, I find a dish pan works really great to float the plants, because they collapse. And if they're in a dish pan, I can see more clearly the structure of the plant. and handheld magnifiers of any kind. I have a microscope, but a cell phone magnifier works pretty well too. And then look for other organisms that are potentially there. I'm sorry. Handheld magnifiers help with zooplankton and to look for other organisms. They're very, very wonderful to see, and they're just doing their thing there on the plants. And then I can't emphasize enough to use several references. You know what? You never know what you might find. There's something there that you can discover. A freshwater jellyfish, which is about the size of the quarter and only shows up for a couple of weeks at the end of the summer, or a rose begonia orchid near the shore in the marsh, just waiting there for you to see. Communicate with fellow lakeshore owners. Always prepare a written report for the Lake Association meeting and then give an oral report and encourage questions, because the people there they don't know about the plants usually. And if you bring some plants or some interesting materials to examine and for them to touch, some people are reluctant to put their hands in the dishpan where I have some plants or something. And then until they do, they just get really excited about it. And the results of that could be more eyes on the lake. Knowledge empowers people, and the enthusiasm can show for the lake ecosystem as a whole. It's not just a place where you recreate, but there are other organisms living there that are important. A view from our deck, and my references here. If you need them for later, I won't leave them up on the screen. So thank you, everyone.
Thank you, Jeri. That was wonderful. And Jeri has a long running program, so she's a great example of working and keeping things alive for a long time, keeping something going. I'm the next presenter, so let me see if I can get my screen up. And all right, hold on. Okay. I'm a summer resident at Peachum Pond. We're there from May to October. And we are an example of a brand new program. For several years, I had wanted to start a VIP program. I'm the president of the Pond Association, but we have a lot of things on our plate, and it always used to just get pushed aside. And summer comes and goes very quickly in Vermont, as you all know. So last year, we really made a determined effort that we were going to get our program started, and we did. Here is a little bit about our pond. We are a pretty small pond, 330.
I can't see your presentation.
Oh, no. You cannot. Okay, let me go back. Let me see if I can get out of here. You cannot see any of it? Okay.
No.
Okay, let me see if I can go to, I'm going to try my other presentation. Hold on. I'm sorry.
If you send it to Beth, could Beth seem to have it?
Martha, I can share my screen if you want.
There we go.
Okay, you want to do that? Because I sent it to you. All right.
Yeah, I think I've got it here.
Okay, let me, do I need to stop my share? Hold on.
Wow.
Okay, is it up?
Yes.
Okay, and I can't see it. Okay.
Hmm.
Dave, will you
Click on your Zoom
slide?
icon,
Okay. So
Martha.
would you go to slide 2 that has Peachum Pond on it?
Yes.
Okay, there we go. I'll just talk through my slides and you can share. I'll tell you when to change. Okay, so our pond is 331 acres, a maximum depth of 68 feet. We are in the Groton State Forest, basically surrounded on many sides by the forest and up in the Northeast Kingdom. We have only about 80 landowners on our pond, which, like Jeri said, it's a little harder when you don't have as many people to find the volunteers. But we have very enthusiastic camp owners. And another thing that's unique is that we are, sorry, I spelled dam wrong. It is a dam. We don't really love it too much, but anyway, we have Green Mountain Power controls our dam water levels, and they're dropped during the winter and raised in the spring. And that we're wondering, that may influence a lot of our aquatic species, land and plants and animals. All right, next slide is my timeline. This is what worked when we needed to start a program. We started early this time of year. I sent out a bunch of emails. We contacted about 20 possible people that I thought, might want to do this work. And we got about 10 of those people who said, sure, let me know what's going on. So in May and June, when the training schedule came out, Kim posted her training schedules. We shared those dates and times, and the volunteers, looked at their schedules to see if they could possibly attend. Of those 10 volunteers, we had 5 who could actually train. So in July and August, we chose an online class, and we actually got a paddle training at Peach and Pond. Julie Silverman came out and did our paddle training. And so 5 volunteers were either trained online or paddle training, or a combination of both. And so we got a little bit of a late start, but during July and August, we divided up the shoreline so that we could monitor most of it, where we knew the most important areas were, at least the shallower areas, the areas with inlets or with water coming in, with our little streams coming in. We used Lake Map, very, very crude compared to some people's. And we set out in August. We shared our data expectations. Everybody was pretty much in charge of their own data, but we did use the online log sheet, and we put in, the time, the date, the weather, what we noticed, where did we see any invasives, all those kinds of things that Kim wants to know. And in September, we added up our hours, and we had actually compiled, with training and logging and identifying our plants, we logged 35.5 hours for our first summer. We were very proud of that, because that $426 is our volunteer match that goes back to us getting our greeter program funded. And so that's an important thing for us. We need those volunteer hours, and that was a great way to earn them. Next slide says, lessons from our first year. Well, we learned a lot. Keep reaching out to a larger group as possible, because you will find someone in that group who would like to volunteer. I think the amount of aquatic plants is overwhelming. So learn what the invasive plants look like first. Really focus on learning those plants, so that if you encounter something that looks invasive, you can, identify that quickly. Work in a team. I like that, just because I think it brings enthusiasm, and it also helps if you have more eyes working together, and maybe at different depths around the pond. Bring samples home to examine. I think everybody does that and says that. Go slow. Don't panic, because it is new, and there's going to be a lot of things you don't know before you find out what you do know. In the DEC has a wonderful plant key. Kim will teach you how to use that during orientation or during training, and it worked really well for us. We could identify almost everything we found using just that plant key. File your reports while things are fresh in your mind. You can do that anytime during the year. And like Jeri just said, tell the neighbors what you're doing. So, because you're going to be poking around their dock and around their shoreline, and you know, people don't always want you up close, but if you tell them what they're doing, they'll be happy to have you there. Next slide is just a few pictures. Here's my partner. And here is our very scientific way we collected our plants. We threw them on the front of the kayak. So next year we'll try to use bags and maybe jars, but you know, we were just learning. And we brought them home, and by the time they got back, they were probably all dried out. But we did, again, could identify most of what we found. Next slide says our plans for next year. We would like to have more viewing tubes. I found that looking for things below the surface was really challenging if you didn't have a tube. We have one, but I feel like every team or every person who's surveying should have a tube of their own. So we'll look into making those or buying those. I'm very excited about learning to use iNaturalist. I know I will be calling Julia, and we'll be setting up a little bit of our own site, I think. So it's wonderful. And I'd like to create something that we can share online as a pond community with our hours. And maybe we can do all that through iNaturalist. We'd like to find some funds to do more mapping or plant surveying so that our pond has a current map of aquatic species, and just build our volunteer group and learn and enjoy. We learned so much in just one short year. It was a pretty incredible. My contact information is on the last slide, and I know I'm not a wealth of information, but I'm really happy to have anybody contact me. Okay, it's been a great experience. Thank you. Thanks, Dave, for saving me. Now we have one more presenter, and then we're going to go to Q&A. Our last presenter is from Echo Lake, and Peggy Stevens. And Peggy is another person who's been doing this for a long, long time. Are you ready, Peggy? See if she's there.
Now can you hear me?
Yes.
It's good to meet you all, and thanks for having me. I sort of feel like saying what they said because, you know, I just learned that I have so much in common with you, all of your experiences. And I also want to thank you just for putting this on today. I've already learned so much, which is my experience every time I interact with the Division of Lakes and Ponds and with FOVLAP. Just that, like, for example, iNatural and GoPro, thank you, David. It's a great idea. And just all the online resources, I already feel like I have gained so much for our program, which started in the early 2000s, I think just behind when Seymour did. You need to understand, Echo is like the little sister of Seymour. We're connected by a brook, maybe a quarter mile between us as the crow flies. Much smaller lake than Seymour, about 500 acres, about 5 miles around in circumference. So it does make it easier for us to effectively cover the entire circumference of the lake in our survey. As I said, I don't know the exact date, but it was the very early 2000s. We started with Ann Bove, and that was, I think, a great way to launch our Access Greeter program. So the VIP program and Access Greeter program basically started simultaneously, because you can't really run an effective Access Greeter program without highly educated inspection staff. So with the advent of our Access Greeter program, in addition to our greeters, we also invited folks around the lake to join in on that VIP training. We first trained 8 individuals, and over time have trained at least 12.5 folks that I could think of off the top of my head. I guess record keeping isn't my strong point, but we have a very active community of VIPs because, as you've mentioned, there's been a little more turnover in the last couple of years, but basically our population has been very reliable. The people who come to ECHO stay at ECHO. And so we've been fortunate in that regard. So in terms of, I guess, the value of the Access Greeter program, because we consider the Access Greeters our first line of defense. And we have, between our paid staff and our volunteer staff, we have pretty much covered 12 hours a day at the Access between May and the end of September. So we grab the first fishermen to launch in the morning, as well as the more recreational boaters. And we're there when folks are leaving the lake at night as well. I think education, you've made that point that the VIP program, the education provided is absolutely the most valuable resource. And we really encourage, as I have heard you saying before me, we've really encouraged folks not to feel like they have to be VIP trained in order to have their eyes on the lake and to get to know their property, their waterfront. And the more familiar they are with the vegetation that's growing right in their front yard, literally, the more comfortable we can feel. I'm the coordinator of the lake bed survey program, and I get a lot of calls throughout the summer, or people coming to my porch with samples saying, can you tell me what this is? So the level of concern and awareness is very high amongst our community, and I think that is probably also a great strength. I don't want to be redundant here, so I'm just going to run through my notes. One outgrowth of the Access Greeter program was the lake bed survey program that Leslie Matthews came to ECHO to do in about, I think, 2007. So we really broadened then our second line of defense, which is the lake bed survey. We use a geological survey map, and we have sectioned off so that each of our surveyors, we have a team of a dozen who survey the same sections regularly. There's overlap between sections, so we know we're not missing anything. Very often we do go out in teams. We minimally are inspecting in late June after established communities may have begun, and then we inspect again in September. But we also are inspecting throughout the summer whenever we go out on the water, either in a kayak or swimming or snorkeling. So there is an ongoing, I would say an ongoing survey throughout this summer. And the fact that we have returning lakebed surveyors who really know their sections has become also a really valuable resource, because we can literally say, let's go see if that robin's pondweed is still where we think it is going to be. So we've been mapping the vegetation over the years, and we can, this I think the most important thing that we're doing right now, we literally know where, generally speaking, where we're going to find certain species of aquatic vegetation. We note where lake bed is filling in. We note where vegetation is becoming more dense. And what we've come to find is that these instances are usually where streams or little brooks are inletting into the lake. And so we have now just gotten a La Rosa Partnership Program initiative started, and it should really help to confirm our suspicions about where runoff might be occurring, bringing in nutrients. Phosphorus especially is of concern, because there's been a trend over the last 10 years increasing phosphorus levels, not yet completely dangerous, but certainly enough to make us be taking a special care. And that leads me then to our latest initiative. We have petitioned our town and the state to have our lake reclassified because we are eligible as an A1 water body, even though we're currently classified like most lakes in the state as B2. We're actively seeking to have that classification upgraded so that we can access more resources to help us figure out why our phosphorus levels are rising, where those point sources for phosphorus and other nutrient pollution might be occurring, and then be able to mitigate those problems. So I guess all to say, you know, since the very beginning in the 80s, when my late husband began doing the water quality survey in ECHO, to then the inception of the excess greeter program, the lake bed survey teams, now the La Rosa project, and hopefully the soon reclassification of our lake, I can look at our history, which this has given me an opportunity to do, and say, wow, we have just developed some wonderful programs and a really strong community here on ECHO that, you know, we've got folks who really care, and that's the first ingredient in a successful program. Caring about your lake quality is going to increase the motivation of your surveyors. So I have no idea what time it is, but I think that's sufficient to give you an overview of ECHO's program.
That was actually perfect timing, Peggy.
Wow.
Thank you. And thank you to everyone. We have some questions in the chat. You can also raise your hand. I think somebody will notice it. From Mark, David, are you using a pole on your camera? The answer, David, yes, with a flotation device on top. Yay. And Amy says, excellent tips to Geraldine. A lot of great jobs. Sweet orchid. Chrissy has a recommendation. While I patrol, she says she carries a quiver of cut lengths of wire coat hangers fashioned into lake bottom markers with a bent loop at one end and tied with survey tape to flag underwater sites of suspected AIS plants. They hold up well and easy to spot. I would love to see a picture of that. And where do we get our viewing tubes? Now, you can make them. There are ways to make them, and I'm sure there's a YouTube. There's a YouTube for everything. You can also buy them online. They're expensive to buy online. The ones that we have at Seymour, DEC actually gave to us. Does anyone else have any other resources for that?
I put directions in the chat box on how to make them.
Oh, great.
And then if anybody wants to borrow one, they can borrow one and from me.
Great.
I suggest attending the lake seminar because that's where I won it in a random drawing. And I love that. And so perhaps that's an idea for the lake seminar.
Yes, and there are directions in there. Chrissy says Peggy has a good point about streams and inlets. It's always good to notice the presence of erosion and buildup. And that's part of the reason, and this is not related to detection of invasive species, but when you do your lake bottom surveys, when you have increase of vascular vegetation in an area, that indicates that there is more nutrient runoff. So that can help you identify non-point pollution sources. So the lake bottom mapping is good for that as well. The La Rosa project is a sampling. You can collect samples and get grants to have the samples analyzed through the state of Vermont. Kim, I bet, has better information about that than I do.
Sorry, multitasking. Yes, the Rosa project is a water quality sampling project. The grants are through, it's not the lakes and ponds, although we are trying to take on that entire project. But if anybody is interested in becoming a volunteer and doing water quality sampling beyond lay monitoring program with Mark Mitchell, then you can contact me and I'll try to provide the right information for you.
Okay, and then we have Ed Wells says small marker buoys used by fishermen work well to temporarily mark a plant location. Amy from New Hampshire says a swim noodle cut into 8 to 12 inch lengths are good floats attached to a 10 to 12 foot rope and weight. Heavy washers or nuts are good. And then once assembled, the rope can be wrapped around the noodle and float, and you can carry them easily in your kayak. All great ideas. And then from FOVLAP, this keeps moving on me. La Rosa is specifically for tributary sampling. And at Shadow, Chrissy says that you got some round orange buoys donated from Ovis. That's great. And then Jackie Sprague provided a link to La Rosa monitoring. So there must be some more questions. Just unmic and ask yourself. Yeah, ask a question.
If people want to raise their hand, that works as well, as easily as typing. So just raise your hand and we'll call on you.
Buried gas marker flags for 20+ years, Jamie says. That's interesting. Some more links in here.
And Dave, whoops, I thought David had his hand up.
Oh, yes.
He does. Go ahead, David.
Hi, is Peggy Stevens still here? She really nailed it on the head, and I wanted to cut my presentation short, but she has, I can't wait to meet her. She is in a lake right next to us, Echo Lake, and she nailed it on the head. For us at Seymour, what I've noticed, or we've noticed, is that we have a lot of growth of our native plants near runoff, and also where the older septic systems were placed, right near the shores. And there's a direct correlation between these septic tanks and a high growth of what was just pipewort in our lake. But I've noticed it. I've kayaked around our lake a million times. Every time you go to the older septic tanks, there's a lot of growth there. What we do at Seymour, we also make sure to mark or mark where our boat launches are. We have 4 or 5 places on our lake that have a launch. So those are obviously also high patrol areas that we take a look at. Yeah, as the nitrogen and all those levels have gone up, phosphorus levels have gone up. There's more growth those summers. And I can't wait to talk to Peggy. I wish she was still here, because she really nailed it on the head. I kind of, she said it all as far as what we're doing on Seymour. And everyone has done a great presentation today. That's all.
Wire flags with invisible dog fencing can work to mark sites. I hope it's not plugged in. It couldn't be, but.
There's a hand up on the phone, person on the phone.
Oh, person on phone, go ahead.
You need to unmute.
Hi, this is the phone person, Valerie from
Oh, it's Val.
Lake Memphis
Valerie.
area. Hi, hey, I wanted to add something to the folks that were spotting the heavy vegetation. Also, keep an eye out for filamentous green algae. I've seen that give me a heads up before the growth of the vascular plants come up, and that's often associated with the runoff or, in some cases, septic systems that are leaking.
Yeah, we've had a great increase of the filamentous green algae on Seymour. And actually, Valerie is a celebrity because she is the VIP person who found starry stonewort in Memphremagog.
Yeah, I just wish that we could have gotten on it sooner because it took about 4 years before they recognized that was what it really was.
Yeah.
And the funny part to that story is I couldn't figure out what it was. I was just, exploring the area, and it wasn't in any of my keys because those keys came out before that stuff moved to the U.S.
Right. Yeah, and if Valerie
But
could.
but anyway, be careful if the thing that we don't talk about for VIP stuff are two aspects. One is, yeah, there could be septic stuff, which means we could be in the water where there's not such good healthy water, and also to be alert for cyanobacteria.
Oh, yes, that's very important. If your bodies are in the water, dogs with you, most lakes in our region don't have to worry about cyanobacteria, but some of the larger lakes, Memphremagog, Champlain, cyanobacteria can be a health hazard. Great point. This is a good resource. Diversdirect.com. You can buy viewing tubes for $30. I like that because we paid a lot more for ours. We got an official one at Seymour, and I think it was $100.
Other questions?
How about inspiration? Does anybody have any inspiration from today? I guess not.
I'm inspired.
Thank you.
Okay, well, yeah, I told my sister-in-law, I think my head's going to explode. I got so much new information today from everyone. So unless something else pops up in the chat box, we're going to start wrapping up and saying our thank yous. Thank you to everyone for attending. Thank you to everyone for presenting. I know Pat and Jack would like to say a few words about completing the survey and other things, but as your moderator, I really enjoyed myself and I enjoy all of you. And one day we're going to see each other face to face, right? Those days are coming again. So maybe I'll see you at the lake seminar. All right. Thank you very much. And here's Pat and Jack.
Pat, you want to go first?
I will go first. I just want to thank everybody for coming here. I want to thank, this was fabulous, I have to say. I learned a lot, so interesting. I'm writing all kinds of notes for my lake and my lake association too, so thank you. I want to especially thank Jack, the leader of our AIS committee, and for organizing the committee and organizing these events. Thank you, Martha, for moderating, and Beth for handling all the question and answer. It was just great. Thank you to all the speakers. Everybody was so great, so interesting, really, really well done. I also want to thank the events committee, Dave Johnson and Jackie Sprague particularly, and everyone else on the events committee for helping to organize this and taking care of a lot of the logistics. Except for my inability to share my screen during the break, which I don't know what happened, because usually I don't have that problem. But anyway, so, and also a special shout out to Jeri, who made a fabulous slideshow for the break that had great pictures of invasive. So we will include that with the other event, the other slideshows that were shown at this event, and it will be there. They're really good pictures and very helpful. So this was terrific. But I don't want you to go away without a reminder to mark your calendar for our next event. So our next event is the Lake Seminar, and that is Friday, June 3rd. And that will actually last longer. It'll be most of the day. The focus is going to be climate change effect on Vermont lakes. And we'll also be doing a little bit about the 50th anniversary of the Clean Water Act, really looking at where we were, where we are now, and now what we're facing with climate change for lakes. So please mark your calendars for that. That will continue to be a virtual event. Unfortunately, we made that decision feeling a little bit unsure about the COVID situation when we had to decide. So still we'll see each other on Zoom again, and hopefully our next event we'll have an in-person opportunity. So once again, thank you, everybody. And I'm going to turn it over to Jack to close the meeting.
So I'm inspired by this, and I hope that many of you are. In our group, our ad hoc group, I mean, this is what we've been hoping for. I don't know if some of you attended our last one, which is how to get your supplemental funding for your greeter program, but we're trying to come up with these timely, important boots on the ground, things that will really help the lake associations. And I'd just like to thank Pat and FOVLAP, because Pat began her presidency back in September, and I think she feels that, as we do, that trying to get to you and the lake associations on your lakes with these innovative, and I would say inspiring events is really what we're all about and what we're trying to achieve. And, you know, we certainly have other room on our committee for people that would like to help us. And one of the things that I think that came out of today is, you know, this will be videotaped. If you were inspired and helped by what you saw, talk to your friends, talk to your families, talk to your neighbors on the lake. Make your life more fun and easier by wanting to do volunteer activities like this that really are fun and give you a sense of together we can manage this and we can all do better. So this is our goal, and I think we've realized it in the meeting, and it's really Martha who came up with this idea and who ushered us through there, and she's really done just a terrific job. And thanks to all the speakers today. These were really terrific presentations with many take-home messages. So thank you all, and don't forget the questionnaire, which you'll see when you close out here.
Jack, we have a couple more questions. One of them is the chat. Does anybody know how to save the chat? Will the chat be able to be seen on the video that's posted?
You don't usually see it on the video, but it should download when we close the meeting. And I made a note to see how we can include that when we share the presentations, when we share the slideshows, to try to put the chat in there.
Great. And I'm going to put the FOVLAP webpage up here, but where on the FOVLAP webpage are we going to be sharing this video?
There'll be a link. We actually have a FOVLAP YouTube channel. It's not well populated yet, but we are building it. So there will be a link on the FOVLAP webpage to the video, which will actually show up on YouTube. So it'll just link you right in. And that might take a few days. It takes a little time to actually do the first, for everything to be downloaded and then to upload it to YouTube. So look for it, wait a couple days, and we should have everything up.
Okay. Pat, Mark Malazza just said, if you go to the bottom of the chat, way at the bottom, there's 3 little dots. If you click on that, there, it says you can save chat right there.
Good morning.
Yep.
Is that
Thanks.
correct?
Yeah.
Okay.
Yeah.
Thanks to everyone for being here. You know, and
Oops, I don't know where that came from.
Okay, is there more? If not, I think just like to thank everyone and close out a terrific session.
Thanks, everyone. Great job.
Thank you.
Thank you. Bye-bye.
Thanks, everyone.
Thank
Bye.
you.
Thank you so much.
Thank you.